it's kind of nice to be reinvigorating this old study because overwhelmingly the response has been great and the people are super excited about having been part of this science effort before and now wanting to continue to do this. So unlike a sort of general population group where there's no buy-in, I think these guys really feel part of something bigger. And because of that, I think they have much more excitement about continuing to participate.
Welcome to My Nuclear Life. I'm Shelley Lesher. On our last episode, I discussed the history and impact of the baby tooth study with documentary filmmaker Nina Gilden-Seavey. As we teased in that episode, the baby teeth from the original survey have been found and are part of a new study led by Professor Marc Weisskopf. Marc is a professor of epidemiology and physiology in environmental health at Harvard University, and his group works in environmental neuroepidemiology. His body of work is impressive and centers around how environmental factors affect the nervous system and the epidemiology of neurological disorders. You will hear more about his work during the podcast. What people find most surprising about this story is that thousands of teeth were found in a closet decades after the conclusion of the original study. If you're in an old lab or maybe even in an old office, this is actually not the surprising part. We all have these little kind of archaeological sites around. My old PhD office had decades of data stacked on shelves in antiquated forms I'm not sure anyone could ever read again. We also had old computer punch cards, which actually did come in handy to stabilize a table or two. I think experimentalists are just pack rats. You never know when you might need that old dot matrix printer cable or a floppy drive. Even in my current lab, I have equipment labeled with the name of a university, which has not existed since 1964. So look around your lab and let us know what you find. I just wanted to start by asking you what your area of research is in general, before we then talk about how you came to the baby tooth study.
Sure. So very broadly, my area of research is, I guess to sum it up, I'd put it on environmental influences on the brain and brain disorders. And by environmental, that sort of meant toxicant kind of exposures, but we're trying to think a little bit more broadly about the environment and things that affect the brain. So, you could go somewhere between toxicants and at the other extreme is sort of anything not genetic, I guess, and how that influences the brain at any point. So we look kind of across the life course. So the sort of outcomes we may look at are autism on the neurodevelopmental end or Alzheimer's disease on the neurodegenerative end or almost anything in between.
And how do you find kind of sample sets, or how do you figure out the factors that are going to impact those neurological disorders?
I got into the idea of the environment in general just because of the sense and thought, and I think it is correct that the environment can influence the brain in different ways, and the brain is susceptible to things like that. We know things from sort of in vitro and animal experiments about very specific things certain toxicants can do. And so ideas can come from there, but ideas can also come from sort of a more public health lens in the sense of there are these exposures out there. Should we be worried about them? Let's look at what we see, even sort of absent an a priori hypothesis about something. Although quite frankly, most toxicants, if put in the wrong place, can do bad things to nerve cells. So it's sort of fertile ground, I guess. After that, it then becomes a little bit a matter of, you were asking about epidemiology, and a lot of this is figuring out settings and data sets where you can actually answer questions, right? So you unfortunately just can't answer every question, and some questions you can answer in a better way than others. So part of what we do is sort of seek out the best setting to be able to answer questions that we think are relevant. And sometimes that means we have to leave certain questions to the side and say, I can't answer that question. Some people who do the kind of work I do, environmental health work in general, are sort of known for, they work on lead. I do a lot of work with lead, but not only lead, in part because I'm looking for the combination of an exposure and an outcome and a data setting that I think I can answer a question. And so sometimes that forces me to move off of a toxicant that I've been studying because it's not a question I can answer. So I'm going to go to a different toxicant and maybe a different brain outcome where I think I can get a better answer to some questions.
Isn't it hard to look at kind of that long-term result?
By long-term, you mean?
So if someone's exposed in vitro and then they get Alzheimer's, isn't that a really hard thing to pinpoint?
Very hard. That's very hard. So there's many facets to epidemiology, but there's no question that sort of the techniques that you use in epidemiology are much better suited in some senses for very acute things. So if you want to look at an epidemic like coronavirus or something like that, or you want to look at a very acute effect of a medication or something like that, it's easier to do. If I worked for 2 years at the CDC as what we call an epidemic intelligence service officer, and we were supposed to go around doing public health work. And the sort of classic training you get in that is 10 people got sick at the church picnic. What was it, right? And you find out it's the potato salad and someone put an old egg in there or something like that. And with an outcome that occurs a matter of hours to days after whatever the exposure is, that's a little bit more tractable in an easy way. That's not every question you get. And so I do a lot with effectively chronic diseases, and those are much harder to deal with because you're looking at exposures that, one, you don't really know when the exposure mattered, and so what time period you're trying to look at. And two, it's often exposures that occur over a very long period of time. They accumulate slowly. People don't really know they're exposed. So that introduces all sorts of difficulties in how to do the studies. But then if you want to move on to this next realm that I'm very interested in now, I guess, unfortunately, because it's a hard question to answer, that early life sets you up for your health trajectory over the full course of your life, even 60, 70, 80 years later. Now you're really in difficult terrain because you've got to know what someone was exposed to 70 years ago, which is often you can roughly estimate, but it's hard to get very good pictures.
Well, and I thought I saw a study that you looked at something that people's grandparents were exposed to.
Right. Yeah. So that's even taking it another level forward. And also that's actually, I should start this by saying this, that's maybe an example to some degree of another set of participatory citizen-based science. So yeah, so there's these interests in, I actually have often talked about my work as kind of toxicant exposures across the life course. And now I have a sort of tongue-in-cheek slide that has in parentheses life courses after that because of this. And the idea is, here's a place where animal experimentation work helps enormously because you can just control the scenario much better. But the idea is you can have effects on cells that affect you. So, a somatic cell that is mine and I could have either keep for life or it divides again in me can affect me. If I do something to that cell, that can affect me over the course of my life. But germline cells are the ones that produce the next generation. So if you affect a germline cell, and it stays, you can, in theory, affect the next generation. And you can also, frankly, mutate a cell, carcinogenesis in some way, or mutate the DNA. And that DNA would then get passed to the next generation. And in theory, the next generations after that for some point. But there's also been, relatively more recently, a lot of interest in what's called epigenetics. So the best analogy of this that I've seen actually is the score of, say, a Vivaldi piece or something, right? And you've got the music that is the DNA. But then the people who are playing it at La Scala in Milan are marking it up saying, go fast here, go slow here, or, downplay this, play this loud. Those marks on the music score that stays the same no matter where it's played, the marks change. And that's the epigenetics. And that's what we all have too. So there's all sorts of ways to effectively mark your DNA to say, read out more of this, read out less of this. It's in fact why a liver is a liver and a brain is a brain, right? Those cells have the identical DNA, but they're told to read out different parts of it. And so it turns out that also is affected by environmental exposures, psychosocial exposures. And really importantly, while some of those can change relatively rapidly, some are not changeable very rapidly and in fact are inheritable. So you can sort of pass on the same DNA with marks from something that your mother, your grandmother, your father, your grandfather experienced, right? So we've tried to look at questions like that. There are very few settings you can do it. And the way we assess exposures are not terrific. We focused on very clear exposures that is easy to ask someone about, like, did you smoke during pregnancy? Or did you take diethylstilbestrol? Or did your mother take diethylstilbestrol when she was pregnant with you? To see whether those things get passed on to the next generation.
Okay, that's just insane that you can pass on those genes.
So I will tell you what's even more insane, which is, there is actually also some experiments in rats, I believe, where they have shown, I think this one was through the paternal line, that if they effectively did fear conditioning to an odor to a male rat at the right time, the fear of that smell was passed on to the next generations. So, the offspring would exhibit the same fear-like behavior to that trained, the one that you trained the parent on.
How did you come across the baby teeth in this setting and in your quest?
So I had done a lot of work on metals of different sorts and neurological function in adults, but also in kids. And it's been known for some while, and there's been this interest in this thing called the Barker hypothesis after David Barker, who was one of the first people who sort of put forward this idea that early life exposures may matter later in life. And he did it specifically around the issue of looking by county in England or the UK at birth or rates of low birth weight and underweight and things like that, and cardiovascular mortality like 50 years later, and said there is this association between those two. We think the birth weight is telling us something about general health of the pregnancy in a very crude way. And so he put forward that hypothesis. It's sort of known as the Barker hypothesis. Other people have looked at things like this. In World War II, there was the Dutch famine when the Germans cut off food supplies to the Netherlands. And that happened over a roughly 9-month period. And so people have looked at births that occurred at different times relative to when that food shortage happened. And you can see various effects on schizophrenia, on obesity, on different things, depending on the timing of when that occurred during the pregnancy. So this idea that very early life sets you up for problems later has sort of been around for a while. A lot of study goes into pregnancy-related exposures and childhood outcomes, but less on sort of much later life outcomes, although Barker sort of started to push this, and there is the Dutch famine winter studies. But so that was in my thinking about, I'm looking at all this stuff of when adults are exposed to lead, does it affect their cognitive function? And then thinking, but is that the only thing that matters? Could it be related to other things? And then about 15 years or so ago, I saw a presentation by an animal researcher from URI who was presenting work showing that he had exposed rats in utero to lead only. And of course, in an experimental setting, he knew it was only in utero. And he was showing that in this senescent rat, which is a couple of years later, you see pathology in the brain that looks a lot like Alzheimer's disease. You see more amyloid beta. You see certain proteins related to Alzheimer's disease that are upregulated. And he was positing that the early life lead exposure had led to epigenetic changes, so not DNA changes, but these marks on the DNA, that led to overexpression of bad Alzheimer's-like proteins in later life and predisposed to dementia. And so I saw that and I said, wow, that's incredible. Right. Too bad I could never do that in humans because I got to wait 70 years and I can't do that. Right. And so that was sitting with me for this long time. Like, how the heck can we possibly do this? And then there'd been several years later to that, there was sort of a few people who started sort of reinvigorating the idea that teeth are a very interesting biomarker of exposures. Now, that had been known for a very long time. And in fact, the St. Louis baby tooth study was based on that idea that teeth accumulate things and we can look at what people were exposed to. And it sort of got back into the consciousness that teeth could be of interest. And in fact, some of the earliest work on lead and the effects on children and adolescents of early lead exposure was from teeth. They analyzed lead in teeth and said that's related to behavioral problems in adolescence.
Okay, so how are teeth a good biomarker?
Basically because they are essentially a fossil, even when they're in your mouth. So they accumulate stuff when they develop, and they develop in a very well-known and defined time frame and in a very defined process, in fact. So we can actually look very closely at the sort of anatomy of the tooth and know exactly when certain parts developed. But that, your baby teeth, for example, start in about the second trimester of pregnancy, and they finish within the first year of life. And it's supposed to be hard to like eat things and chew them down. They don't change pretty much. I mean, they can get worn down a bit, but the certain parts of the tooth, they're done. And so once they form, that's it. And that's why physical anthropologists, when they discover, Lucy in the Rift Valley in Africa, will look at the teeth, because the teeth are, one, they hang around a long time because they're sort of like stone fossils, but they can look inside them and say, well, okay, what was the diet like of this person 1,000 years ago? So we can do that with any tooth if we get one from somebody and say, we know from the second trimester of pregnancy to early first year of life what was circulating in the blood and what got deposited in the tooth while it was being formed. An adult tooth forms a little bit later, but you can basically do the same thing with the adult tooth. And so it's a very unique biomarker in that it gives us like this snapshot of a very well understood time period of life. Right. We often, when we do environmental health work, we're looking at blood, what's in blood. We're looking at what's in toenails or hair. Or I do a lot with lead in bone, which is also a pretty good one because the lead and other metals get incorporated into bone. And bone turns over, depending on the bone type, very slowly. So it accumulates lead. The sink for lead in the body is the bone. And so 95% of anyone's lead is put in the bone, but it still turns over somewhat. I mean, people get osteoporosis in older life. In menopause, women lose a lot of bone. So it's not as good as teeth even because it does turn over and can be influenced by later things. But it is a pretty good one. But you can't go back to childhood with a bone because...
Well, and it's hard to remove a bone.
It's true. So removing a bone is harder, but there are actually nice... Physicists have come along and said, we can figure out how to determine lead in a bone without actually having to cut it out. So we can use X-ray fluorescence, which is essentially, it's totally non-invasive.
Okay.
It's an X-ray. It's effectively like an X-ray of the bone to determine lead levels.
And so you have this timeline of a tooth that tells you what the person was exposed to before they were born, and then you can tell when they were born and then after?
Right. So if it's the baby tooth, which starts in utero, teeth grow on a daily basis, little like trees. So, tree rings that have an annular, every year you get a ring in the tree. And so you can sort of cut a tree, not if you want to do that, then you can sort of count the rings and say, how old was this tree? It's the same thing with the teeth. So every day they lay down a layer of cells. And with the proper cutting of the tooth and visualization, you can sort of see those lines. And one of the things that happens is if you have stress periods while the tooth is forming, that affects the laying down of those cells. And one of the biggest stressors in early life is actually the birth. And so at the time of the birth, it sort of slows the laying of these cells down and creates a much thicker line that actually is much easier to visualize than the other daily ones. So we can sort of cut a tooth in half, visualize it in the right way under a microscope and say, okay, there's that line. That's when the birth happened. And then we can analyze, the part of the tooth before that and the part of the tooth after that and say, this is what was happening prenatally and this was what was happening right after birth.
Wow. So were you looking at teeth before you discovered the baby tooth study?
This idea of early life and dementia later in life being possibly related, I had started to look into the ideas of teeth. And the first ways I'd gone about that was to try and find like Alzheimer's wards or dementia wards where you had inpatient people with the idea that they're eventually going to die because it's inpatient, and that we would grab a tooth either if they had to have one extracted for some other reason or on autopsy. At the time of death, we would get permission to take one of their teeth. And then you've got to find people who don't have dementia who are willing to give you a tooth. So we were working with a VA setting actually, where they had an inpatient dementia ward and a dental clinic on site. And the idea was veterans who come in to have a tooth pulled will grab their tooth, and then the dementia people will grab their teeth. And it's adult teeth, so doesn't go in utero, but it goes pretty early childhood to look at that picture.
So when do your adult teeth form? Then? Like, what's the time?
Yeah, they start around birth. It depends again on the tooth type. So different teeth, slightly different timings. Wisdom teeth are much later, well, a few years later. So they're a little bit of an anomaly. But the other teeth are basically around the time of birth to 5, 6, 7 years of age. But the problem in those settings was it was still getting very hard to get many people with dementia cases and even non-cases in this VA setting. So we were having a really hard time getting large enough numbers. And a lot of the problems in epidemiology and environmental health come from difficulty assessing exposures and having error in that exposure. And so the one way we deal with that is to have larger numbers. And so having small numbers is always, an epidemiologist is never totally happy about small numbers. So I was struggling to get enough numbers, and then really sort of by chance came across the St. Louis baby tooth study.
And how did that happen?
I was talking around with different people who did tooth metal analyses and in the context of the other stuff that I was doing. And one of them said, if you're interested in teeth, you ought to talk to this guy down in New Jersey who has thousands of teeth from this thing called the St. Louis baby tooth study. And that was the first I'd heard of it. I was like, what's that? And yeah, sure, I'd love to talk to him. So let's see. And then it all sort of snowballed from there.
And how did a guy in New Jersey get teeth from St. Louis?
I know. I'm not quite sure how he came into possession of the teeth. What I will say is another sort of odd chance happening was, the St. Louis baby tooth study back in the 50s and 60s collected nearly 300,000 or maybe a little more than 300,000 teeth from not as many kids because they gave more than one tooth. But apparently like 15-ish years ago, somebody stumbled on a closet in WashU where the original study had been based and found 100,000 of these teeth. Literally opened the door and it was like in shoe boxes, like hundreds of shoe boxes of these teeth collected back in the 50s and 60s. I need to ask Joe. I don't know why this guy got in possession of them, but at least he did. And some people had the foresight not to just throw the teeth away, but to save them. And he was that person. And he literally had them in his garage in New Jersey. And I think knew that they were probably really valuable in some way, but didn't really know what to do with them. And so when I came along and said, I have some ideas, we could do some work, he was very excited. And I don't think he was used to the pace of science and NIH funding. So it took many years to actually get funding to start the process, but it eventually happened. And so he's very excited about it.
That is the good thing about an academic setting, is that the teeth were still there. Because academics don't throw anything away.
Exactly. Right.
It's like,
that's right.
I don't need it now, but someone might in the future. So we'll just squirrel it away and perhaps someone will stumble upon it and need it.
I know all of those 5 1/4-inch floppy disks will come useful at some point. So I'm keeping them.
Exactly. Exactly. And the other nice thing is that teeth don't go bad if you just store them in a closet.
Right. Exactly. So that's a huge benefit of this, is that because you're right. I mean, and we, all sorts of people collect tons of blood samples and whatnot and spend a heck of a lot of money storing them in minus 80-degree freezers and all this stuff. Teeth can sit in a closet. They're pretty easy.
So it was just kind of a stroke of luck after another for you with these teeth.
Exactly. Exactly.
Had these teeth been analyzed before, do you know, or were these just kind of the extras?
So, yeah, these teeth, the ones that he actually had that we're using were extras. They were not analyzed before, because the old ways of doing this, they would have ground up the entire tooth to determine radiation levels in the tooth.
So what was the next step then? So you had to get the NIH funding.
Right. This was pre-electronic days, so there was no electronic file of who was in this set of teeth. And going through 100,000 teeth one by hand is an enormous amount of work. So we first did a little pilot study where we said, can you just get a couple thousand and go through the first couple shoeboxes you have and give us the names? And he had actually done a little bit of that himself and had done something small. But I said, if you can get me a list of a few thousand of these people, because the next step for me was, well, okay, we have all these teeth. Can we find these people? Like, can we see where they are and actually contact them? And so for getting any kind of funding from NIH, that was the key thing we were going to have to show, is that we could still recruit these folks back. So we got, with Joe, we had a list of about 3,000 of these folks. I think we were able to find some kind of address by going back to voter rolls. We focused initially only on the men, with the idea that women's name changes would just make it a lot harder to find the women. So we focused on the men, only looked at voter rolls in Missouri and Illinois, with the idea that probably most of them would still be in that area. And just on that, we were able to find about some kind of contact information for about half of them. And with one letter to the address that we found, we got about a 33% response rate of people answering a brief questionnaire on their life. And I was like, that's unbelievable, because just asking anybody to do a study, 33% is in some cases quite good. And here were people we didn't even know whether we were on the right person or I would have the right address. So to me, that was amazing. And that really got it started, because then we said, okay, we can do this. We can find these people. And that was with very minimal of attempts, through voter rolls and only in Missouri and Illinois. So
give me a sense for what this garage looks like. I mean, what does 100,000 teeth look like?
So somewhere we have a picture. We had Joe take a picture of his garage as they were doing this, but it essentially is nondescript garage with stacks and stacks of old shoeboxes. And in the shoeboxes, each one probably holds a few hundred little manila envelopes with one tooth in each envelope, paper clipped, now kind of old, rusty and falling apart at 50, 60 years later, paper clipped to a 3x5 index card that had the name of the kid, the birth date, the city they were in, the parents' names, a few other details on there. So it literally totally nondescript garage with stacked high with, 10, 15 layers of shoeboxes. So
not that many shoeboxes in the end.
Yeah, I guess I'd have to do the numbers. I can't remember. I think you said there's probably a couple hundred in each shoebox, and somehow do the math up to 100,000. So I don't know how many shoeboxes that is, but it's quite a lot.
Yeah. Wow. And so after you got 33% response rate, just for the men, you applied for the NIH funding. And what were your expectations? What is your kind of hypothesis? What are you looking for then? You're looking for kind of dementia or Alzheimer's?
Yeah. So we focused basically sort of on the same idea that animal experimentation work had done. And there's plenty of other work also sort of looking at metals and cognitive function at different times of life, and both endogenous metals and exogenous, meaning not essential to the body in any way, like lead is one of those. So there is sort of this different literature, but we were really sort of trying to mimic that animal experiment, saying if we knew what you were exposed to in utero and you were exposed to more lead, or we could then expand to other metals, would you have this? We couldn't do neuropathology because we're not going to get their brain to see whether they have these protein changes. It's also in this setting, we basically couldn't do frank dementia and Alzheimer's disease, because if someone had frank dementia and Alzheimer's disease, they're almost certainly not going to respond to our letter, right? And to join the study. So the sort of precursor to that is basically cognitive function. And so how are they performing cognitively? And our idea is to track them over time to see what their rate of decline in cognitive function is. There are many reasons you perform at any individual level. So that's not the best way to assess this. If you're not looking at frank dementia or pathology, the best way is to look sort of how you change over time in your cognitive function. And dementia is basically people who decline faster than we would like. So we essentially said we can find these folks. We'll try and get as many as we can. And we thought we were going to get thousands based on the 33% response rate of the pilot. We were still very unsure about women in terms of being able to find them because of name changes. But even if we only had the men, and we thought we'd get many thousands who might respond to this, they're going to be spread across the whole United States. So this starts to look a little bit like some other big cohorts at Harvard that people have worked with for years, like the Nurses' Health Study, where they recruited nurses back in the 70s, depending on which cohort, and have been following them for years since, but they're all across the US. So you have to find out ways to contact them, and you can only assess certain things. Questionnaire is the main thing. You can then sort of back it up with medical records and things like that. So we were sort of following that model, but there's been a lot of advances in assessing cognitive function in different ways. So, the gold standard would be come into a neurologist's office for 8 hours and go through a series of tests. And that was clearly off the table here. We couldn't do that. But we're working with someone who's a cognitive neuroscientist who's at McLean Hospital in Boston, Laura Germain, who has developed this whole online process of assessing cognitive function and different tasks. So we're partnering with her, and we're inviting people to go online, do a survey online, and then run through this battery of cognitive tests. And so that was the fundamental initial question is if you want to distill it to the base, one simple one is more lead exposure in early life based on the teeth and worse cognitive function later. Do we see that?
Are you looking at other metals? as well? Like just kind of whatever shows up?
Yeah. So, I mean, there are some where we have some a priori ideas. There's some interest in iron status and how that might affect things. You know, there's work in kids on manganese or cadmium. These are known to be toxic metals. And so if they are higher, you know, that might be some problems. But there's also a lot of interest in environmental health now in the idea that we're not exposed to one thing at a time, right? In a lab, you can do that. You can just dump only lead on and see what's the effect. In the real world, you're exposed to a mix of stuff. And so there's a lot of biostatistical work on how do you take multiple exposures that are often correlated and sort of find patterns of, okay, if you're high on this set or this combination and low on this, you have associations with some outcome. And so we're also going to be applying those kinds of statistical methods to say, if you sort of take a broader picture of the whole set of these metals, are there patterns in there of certain, maybe it's not just lead, but it doesn't matter. You're high on lead or you're high on manganese and high on either one and you have a problem. Or maybe if you're high on both, that's doubly bad than just having each one individually, things like that.
Do you have to destroy the whole tooth now, like they did in the 50s and 60s?
Right. No, we don't. And that's one of the beautiful parts of this is that we can sort of keep it. We do have to cut it in half and then you can sort of see the layers of the tooth. So you can see the enamel, you can see the dentin underneath. And we actually wind up cutting it in half and then making a sort of thin slice of about 70 or 100 microns. And we ablate with a laser very fine spots, so 25, 50 micron spots to go across essentially the time of formation of the tooth to look at over time what was happening in that tooth. But that leaves us basically with the two halves of the tooth. So we can still do other stuff with those teeth.
Fantastic. So with these 100,000 teeth, how many are like, what was the first step in trying to find everyone? Like it must have just been horrendous trying to just catalog everything.
There were so many, I mean, I knew it was going to be a lot of work. I was still, despite that, surprised each time that new things that caused more work were coming up. So, I mean, I knew that one of the major pieces was we literally needed someone to sit in that garage and with a laptop and sort of transfer all the paper information to electronic information. So we had names.
You couldn't move the boxes into something air conditioned for that person?
That's a good question. I have to say, I wasn't down there. Joe found somebody to do this. I don't even know the air conditioning status of that garage. So maybe he was sweating away. It is true. It is true. I feel a little bad about that, but
I mean, shoe boxes are easy to move.
It's true. I will say that I think this guy had other summer employment, so he did it mostly in the winter. So maybe the concern would have been being too cold in the garage.
Okay,
I'm not sure.
okay.
But so that in itself was more than a year-long process to do that. Now, he would send us chunks of it so we could get going trying to find these people. But it wasn't even like, okay, he sends us a file and we try and find the people. Because when you're hand entering that much information, one, he may make a mistake, put an S when there was a Z or leave out an E or something. So you didn't have perfect name matches. And then, of course, when the people were writing down back in the 50s and 60s on these 3x5 index cards, not only could they have made a mistake or have something that the handwriting was hard to interpret, but you could have come in and said, you know, any one person could have given 3 teeth and probably at different time periods because they fall out at different time periods. So one of those time periods could have been Robert, and then it could have been Rob, and then Robbie or Bob or something. So, you know, how do we know that, you know, now we have the parent's name, but the same thing could have happened with a parent, that it's Ed or Edward or something. And they might have moved addresses. So it turned out to be quite a task to sort of decide who was the same person, right, in this whole file.
So this sounds like a fascinating project for someone interested in genealogy.
It's true. I mean, genealogy and sort of computational programming, fuzzy math, fuzzy linkages and algorithms to decide what's correct. And thankfully, I actually wound up hiring an RA. I had a sense of this, and I think it really did pay off, is that I hired someone who was just out of college who had done engineering. And I think it took that kind of a mind, you know, to sort through this. And she was unbelievable. And she sort of, I mean, I don't even want to look at her computer programs that tried to sort through all of this stuff. But I think she took it on as a great challenge and actually really enjoyed it and did just an absolutely phenomenal job. I mean, and you know, we wound up with We had 100,000 teeth. We sort of estimated based on what she was doing that there probably came from about 36,000 individuals. And she was able to more or less identify unique individuals of about 30 to 32,000 of them. And now we have this pot of maybe 4,000 to 6,000 people where we're a little unsure. Like there are some people in there that look like they might be duplicates, but they might not. But a 30,000-ish that she was able to basically say, yeah, this is a unique person. And we could start with that list. And the next thing is that now we're not just doing voter rolls. We're going across the country to these sort of services that rely on electronic information, like all the electronic footprint we leave in our daily lives. Unfortunately, we can be tracked, and there are companies that do that. But they're doing it based on information that was decades before the electronic era, the internet era, right? So we had no idea whether they were going to actually find these people, given that all we're giving them is their name, date of birth, and where they lived when they were a kid, right, in the 50s.
Well, and you don't even have their mother's maiden name. You just have their mother's married name.
Exactly. And of course, if they're a girl, we don't know whether her name changed since there. And maybe the man, a man too, but we assumed that was going to happen more with the women. Turned out the services did a phenomenal job of getting something. Now, we have to say they give us some potential contact information, but we still don't know whether that's correct, right? So we are sending out thousands and thousands of emails and now letters and, hoping one of them is right for a given individual. But so far, so good. We've had a great response rate.
So that's actually how I found you, is that I was in my dean's office and he had gotten your email because he was part of the survey when he was a child living in St. Louis.
Right.
So he had forwarded me the email saying, the baby tooth study, like I was part of it and now they've asked me to fill this out, and he's retiring this year. So he will now have time to fill out your survey. Excellent.
Great. So that's what we like. People with lots of time to do our work, do our study.
How many unique individuals have filled out the survey so far?
So I think so far we're up to about 2,500, but we've been largely focusing on emails first because it's electronic and it's easier for us. And also people have to kind of go online to do our survey and our testing. But we have a process. And so if this service returned an email to us, we're starting with emails. They also return hard physical addresses and occasionally phone numbers. And so our process is to work through the emails first. And then if we go through all the email, and sometimes we get 3 emails for somebody, potential emails, so email addresses. So once we mail all of those, if we haven't gotten any response from somebody, we'll move on to the hard address letters. If we have no email information for someone, we only go to the letters. And so we have sent out some letters so far. And what we've generally found with this group is that when we send to a hard address, we have a much bigger response rate. But so far, most of our attempts have been through email. We are literally just now sort of have worked through all the emails and decided, okay, we're now at the point where if we haven't heard anything from someone with all our emails, we'll just move on to their hard address. And so we're literally sending out about 40,000 letters. And so we kind of expect a much bigger bump now because we've already, in the few letters we've sent out, we see a much better response rate. So we're thinking we may get many thousands over the summer to join.
So what's the age range of the people? Like the current age range?
Yeah, it's forget exactly. I think it's centered right at around 63 or so. So it's kind of, I think, in the 55 to 70 range, but really focused right around the early 60s.
Okay. Yeah, that makes sense, especially if you're going up to 70, that these are people that may not have current workable email addresses.
Right. And we have had a few people who have said, can you send me a paper questionnaire, which we're trying to produce to send to them. I mean, they can't do all of what we want, but we would like to have them anyway, even if they can only do a paper version and keep track of them.
Yeah, that makes sense. Is there a point in which you're going to start analyzing the teeth of these people? Like how many people, okay, you hit 5,000 people, then you're going to start analyzing their teeth, or kind of what's that point?
Yeah, I know. The ideal situation would be we had the entire cohort of people who were going to respond, and then we'd sort of pick from that. But it's this sort of moving process where it's growing all the time. But for many reasons, scientific and also just funding wise, we can't wait to the end. We need to show we're doing something. So after we got the first couple thousand, we did randomly select 200 to analyze their teeth. So totally randomly from that group. So we've done those. We actually selected another 50 we're about to analyze that also were selected randomly in that group. And that was in part to make sure we're seeing things in the teeth that we might expect. There isn't anything weird. We can do some preliminary analyses with those 200 to 250 teeth, but we're sort of holding off on the rest for this bolus right now. So I'm expecting with the email campaign and mailing campaign now that by the end of the summer, early fall, we'll have a pretty good idea we'll have a much larger number now. And we can, even though people will still keep trickling in, I think by the end of the summer, we'll decide on our final selection process. And part of the reason we want to wait is that we're trying to do something to be a little bit more statistically efficient. You could randomly sample from the whole group, which is great. And that's what we've done so far. And that gives you a general distribution. But if you really want to see whether something is related to a particular outcome, in this case, worse cognitive function, it's more efficient to sort of oversample from the tails. So from people who score really low or score really high. But you don't really want to do that until you see your full distribution. So we're kind of waiting for the end of the summer, early fall to see where we are in our total numbers and then look at those tails and then select oversampling those tails.
Have you looked at the results yet? Or are you waiting?
We've looked at the cognitive results and we see what we would expect. So it's looking good. There's no weirdnesses there. I mean, we see a nice distribution unfortunately, and your dean may experience this when he does this, is the idea behind cognitive tests is to kind of do them until people can't. So they're a little bit annoying in that they're sort of pushing you to the point that, you struggle. And that's the point. We're trying to see where that is. But because you do that, you get a pretty good distribution. You get people who, nobody does perfect on all these things because they're pretty hard to do, but you get a wide distribution. And that's what we're seeing. I will say that this group, at least so far, and maybe people who respond early always tend to skew a little bit more educated, a little bit higher SES. I think that may be the case in general with this population, even if we had everybody. And so maybe we're getting a slight skew anyway, but overall, that's the one thing we do see is that they are a little bit skewed to that end of the socioeconomic education spectrum. But that doesn't mean we can't still see variation in these outcomes, and we can sort of look at those. So we are, and we do have the full survey data on everybody. So we're going to do initial work looking at not the metals in the teeth, but looking at some of their early childhood characteristics that we were asking about and how those relate to later cognitive function or mental health questions.
The survey's quite long from what I heard, like an hour or more.
So certainly doing both the survey and the cognitive function, probably easily an hour. I think we estimated the survey could be 30 minutes on average. It really, though, depends because some of the questions sort of will go on and on. If you've got a lot of stuff going on, that some people may say, nope, none of that. And then they go through it more quickly. Others had more things happening in their life that will make the survey take longer. And the cognitive testing process probably takes 30 to 40 minutes to go through the whole thing.
Okay, that's not bad because I'm part of a large group study, and every year it's like 45 minutes, and I think this year I just didn't take it.
I can't do it
because it was just too much time.
Well, this is a big struggle with this a lot because, as we're developing the survey, we're balancing wanting to know everything and knowing that if we go too far, we're going to get people dropping out because they don't want to do it. So, I will say our intention was to keep it to roughly a half an hour. I know some people have said it's taken me a lot longer. I think there are some people who it doesn't take longer, but that's always going to happen a little bit. But our intention is to, once we've got the final group, is to go back to them every few years and give them another. So we can keep it, hopefully we'll try and keep it on the slightly shorter side, knowing that we can come back 2 years later and ask some more questions. So.
And probably every other year is better than every year.
And the other thing from our perspective of cognitive function is that, 2 years is actually not a lot of time to see change. So we might even go over 3 years or something.
But then if you wait too long, you risk losing contact with people.
Right. Now, I will say on that score, it's kind of nice to be reinvigorating this old study because overwhelmingly the response has been great. And the people are super excited about being having been part of this science effort before and now wanting to continue to do this. So unlike a sort of general population group where there's no buy-in, I think these guys really feel part of something bigger. And because of that, I think they have a much more excitement about continuing to participate.
So have you heard from a lot of these grown children of the study?
Oh, absolutely. And this is where another problem, I mean, it's a good problem to have, but it arose in that we do have to be careful if somebody wants out and they say, I don't want to be part of this. We have to take them out of our database and not contact them. And we got such response. We got so many emails and so many calls and people wanting to talk about the study, tell us what happened when they were kids and all this kind of stuff, that it slowed us down a lot because we had to go through all of those. One, to check just to make sure someone wasn't calling to say, stop contacting me. But sometimes it necessitated an hour-long conversation with somebody, right, about their history in this study. And they were so excited that happened a lot. So there were a lot of people to talk to. So we had to pause actually and change our system to sort of drive them to a website to help us collate when they didn't want to participate so that we could track that without having to go through all the calls. We're still going through all the calls, but we don't have to hold up other parts of our study while we're waiting to do that, number one. And number two, we've sort of created a website with frequently asked questions and all this kind of stuff. So that, because they're so often, I mean, a frequently asked question was, is this real? I don't believe it. It's spam, right? I can't believe you would be doing this. And we're like, no, it really is real.
I did see that frequently asked questions and I was surprised by some of them. So I figured you must have been getting a lot of calls because there's a lot of frequently asked questions.
Yeah, no, we got a lot. We definitely got a lot of calls. Which were the ones that surprised you the most, or were there?
Yeah, I was going to say, can I send you my current teeth? Or
right? Yes, yeah. We're actually looking into that. We're just making sure we're okay from a human subject's perspective to be able to take people's teeth. But yeah, people have definitely done that. People have said, I mean, we've had things like, just, can I send you my own tooth? We had one guy call and say, actually, I have sort of an impacted baby tooth that never came out. Can I take it out and send it to you? So I said, sure, that would be great. Others have called and said, I want my grandkids to participate in this kind of thing. Can I send you their teeth? We want to take them just because we love the enthusiasm. We'll have to, how much we could do with them will depend on sort of how many of these we get. But
well, you certainly won't be able to look at their grandchildren's cognitive function when they're 70.
Right, but maybe I can be the Barry Commoner of this era and set something up for someone 60 years later to do this.
Perhaps. I mean, do you find that frustrating with your work, that it's such a long time scale that you're working with?
At times. I mean, that's why we try and find these shortcuts in a way. I mean, this is why this was a sort of amazing experience to have this sort of fall into my lap in some way, because it allowed me to do these kind of questions. Yeah, it's frustrating, but it's also like, team science in a sense. I mean, we clearly need to do things that we can answer now and get our questions now, but we're also thinking about, my trainees will be around for many years and their trainees will be around. So if we set something up that someone can do stuff 60 years from now, that would be amazing.
Wow. I mean, that is quite a legacy, though.
Yeah, it is.
What do you see the drawbacks of this study, the new study that you're doing, or the potential pitfalls?
Yeah, well, I mean, so by pitfalls, you mean of sort of the questions we're trying to ask, or the science process, or which pitfalls?
Yeah, anything like using the teeth, the time that has gone by, which I think you see as a positive, but what are the potential negatives that all the time has gone by?
Yeah, I mean, so there are definitely negatives. I mean, some of it is just, in an ideal world, it would have been great to track these people since they donated the tooth and really get, as it's happening, information on their lives so we know what's happened in between. We can ask about a lot of this stuff and we know things that are relatively important for the kind of questions we're asking, but still, it would be better to have had that in the past. But if we'd had that, we probably wouldn't be around to answer the questions we want to answer. So there's trade-offs there. I mean, one of the biggest things is just knowing other stuff that was happening around the time when they were kids, because we can get a very good measure of the metals exposures they had. But it's also true that other things come along with metal exposures. People who have higher lead have different socioeconomic status scenarios in their childhood. And so we have to think hard about the kind of questions we ask about their early life to get as clear a picture of that other stuff as we can, but we don't have real-time data on that. And that, so that's a difficulty that we'll have to face and try and handle. in some ways. So that's the most difficult part. But again, it is a trade-off between doing that and actually being able to answer this question with a individualized marker of exposure from so many years ago. I mean, that's so outweighs some of the other problems that we think we can handle the other stuff.
When discussing kind of the socioeconomic status of the children who donated their teeth, do you think that there were a lot of kind of lower socioeconomical children that donated their teeth? And then are you going to try to search those kind of children out for this study? Or can you?
Yeah, so from what we've seen in our data so far, there are, but it does skew to some slightly higher socioeconomic status. But as children, we've asked a bunch of questions about scenarios of their life in childhood. And what we see is we definitely have a spectrum because it's skewed. So that's one of the things when we select out our people we're going to analyze the teeth on, we're actually not just doing it on the tails of the cognitive distribution, because if you do that, you can see that the higher tail is very heavily skewed to more education, higher socioeconomic status, and the lower tail is very heavily skewed to the opposite. So you can't have those differing dramatically, or the comparison is now not just lead differences, but also education and socioeconomic differences. So we have to actually do sort of stratified random sampling where we are going to wind up over selecting people who happen to score low and came from better socioeconomic statuses, because that's rarer, but we're going to have to seek them out to make sure we get enough of them in our sample. And at the higher end, we're going to have to specifically seek out ones that maybe have lower education, worse socioeconomic status as kids, but still are scoring high on the cognitive test. So we have to sort of try and balance some of those things out when we make our selections.
Wow. So I had a question for you, but I think you just kind of answered it for me, which is kind of as a member of the public, how am I able to identify kind of an epidemiological study which I can trust?
Yeah. So this is where I don't know that you're going to love my answer, because as I've mentioned to you before, I mean, I'm fond of saying to some degree that, epidemiology is easy to do in many ways. It's also very easy to do badly. And one of the problems is it's on the face of it, it's very easily interpretable. And the problem is often the media don't have a good grounding in epidemiology and all the issues. And the headlines are very easy, right? Eat strawberries, or, don't eat potatoes and you'll have better health.
Cell phones cause cancer.
Cell phones cause cancer. Very easy, quick headline, right? That just masks so much other stuff. And the fact of the matter is, it is hard, even if you went to the papers without the sort of grounding in epidemiology, and even with it, I will say, I mean, I do a lot in sort of methodology in epidemiology, and I will say that I think there are people who are in the field who don't always get all the issues that are going on, and you can have biases creep in that you don't recognize. There are levels of this. The fundamental thing is you got to sort of look at what are they comparing? People who use cell phones and people who don't, right? So what else is different between those people? And have they tried in any way to account for that? Do they recognize that there are differences? And do you believe that they've addressed them in some way, those other differences? There are some rules of thumb, like oftentimes when we do studies, it's easier and faster and you can get a quicker answer if you go to someone with a disease. You go to people with cancer and ask them, did you use cell phones? And you go to people without cancer and say, did you use cell phones? That's a quicker way to answer a question, and done correctly is very efficient and can get you an answer fast. But it's much more prone to biases. Like someone with a disease will often think it's related to something and they may report differently, right, than someone who doesn't have the disease. Their biology may have been affected by the disease, and that affects what you measure in them, or it may affect their behaviors. And a classic example of this is there was a very well published and cited and influential paper suggesting a very strong relation between energy intake, meaning your diet and how much food you took in, and Parkinson's disease. More food, much higher risk of Parkinson's disease. And it was done in the manner I just told you. They got a bunch of people with Parkinson's disease and said, what were you eating? And then they got a bunch of people without and said, what were you eating? But then when someone came along and instead followed people over time and tracked their eating and then saw who developed Parkinson's disease, they saw no relation whatsoever. And what's known is, one, your reporting of your diet is heavily influenced by what you're eating now. And that when you have Parkinson's disease, your diet changes for biological and behavioral reasons. And so that was a totally biased finding. So a rule of thumb is we tend to prefer studies where you take people and you follow them over time, but you start with people before they have any outcome that you're concerned about, and you follow them and then you see who develops the outcome. That helps a lot in getting around a lot of biases, but also biases can creep into that too. And that's where it gets really nuanced. So it's hard for someone who hasn't been steeped in this to pick it out. And so for the lay public, I would to some degree trust sort of summaries by respected organizations of the larger literature. Even as an epidemiologist, I would, even if I thought one paper was really good, I would not assume causality or absolute certainty based on any one paper, no matter how well it was done. And so often what you read in the headlines is based on one paper that just came out. So at the very least, I would look for more National Academy of Sciences or other large organizations who have sort of summarized something. That said, they can make mistakes in doing that too. But that's, you're trending better if you focus on that.
At least you gave me a starting point.
That's what I would say, yeah.
I mean, usually I just look at sample size first.
That's a big thing too. Sample size and that study design aspect of sort of starting with people without an outcome and following them. Those two things, if you can get into the paper and say those happen, those are major advantages. I love the methodology stuff that sort of gets into the weeds of how even in those settings, you can wind up with some important problems. As an example, there's a really interesting relation between smoking and dementia. Or let's take hypertension and dementia. That relation is, if you look at a bunch of people who are really old and you look at their blood pressure, there seems to be this relation that the higher the blood pressure, the less likely you have dementia. But if you take them when they're mid-age, the blood pressure is related to more dementia, higher blood pressure, more dementia. And there was a lot of questioning of this. I mean, one possibility is when you get really old and blood pressure is an issue, brain requires a lot of blood. Maybe hypertension is good because it's forcing the blood into your brain. But another important aspect is actually that you're looking at a different population. So if you start when they're 70, the people who had high blood pressure before and had problems from that are probably not in your study because they died or because they had other health problems and didn't get there. And so you're looking at a very different group. So you're looking at people who are probably somehow not as responsive to the blood pressure problems, or their high blood pressure only recently started, right? So these are issues that creep in too that, can be very hard to pick out if you're not sort of thinking about these things all the time.
I'll just ask you.
That's fine. You can come to me.
I'll just send you an email like, can I trust this study?
Sure.
Well, thanks. Yeah, that's not a
definitive answer, but I apologize.
No, but it's science, and it's also like things that have to do with people. And when you're dealing with people, it's never
it's
definitive.
Never clean or definitive. Yeah, exactly.
No. So I'm okay with that answer.
All right, good. It's all also, by the way, it's all sort of probability. It's like shades of probability as opposed to like a yes/no answer almost always.
Thank you for listening to this episode, and thank you to Marc. If you think his name sounds familiar, it's because his grandfather was Victor Weisskopf, a theoretical nuclear physicist. Not only was he a great physicist, but he stood by his conscience. While he chose to work on the atomic bomb, he refused to work on the hydrogen bomb. Not just that, but he was a vocal critic of Edward Teller and even dissuaded Hans Bethe from joining the H-bomb project. He really deserves his own episode. But in the meantime, Marc was gracious enough to tell us some of his personal stories about Vicky as they knew him. And you can head over to our website at mynuclearlife.com to find out how to access it. Until next time, I'm Shelley Lesher, and this has been My Nuclear Life.